All right, in chapter 10 here, we're going to talk about bacterial diversity. So this is the first lecture in a couple that are going to look at the wide range of microbes that are out there. Now, I have to get a little bit of housekeeping out of the way here first. Some of these videos are going to use an older naming scheme. Recently, scientists have renamed some of the groups that we see here. Now, if we look at this phylogenetic tree here, we have eukaryotes here, you're in there somewhere, archaea here, and then we have this bacterial tree here, and this is how they're related to each other evolutionarily. Now, we try to group microbes together, and we give those groups names, and over time, things have been named. And sometimes we decide that we need to clean things up a little bit and name them a little bit more sensibly, because sometimes we do it haphazardly. So that's what's happened here. You'll see several names, like the Bacillota, and these are the new names, whereas you'll see the former names. So if you see F. Firmicutes, that means formerly they were called the Firmicutes. That is the nomenclature that's here. In this section, 10.1, I'm going to go through the new naming, but for Micro 111, it's not super important that we follow this new naming. What's really more important is we understand the different groups and some of the diversity of microbes here. So in the following sections, I may use the old naming scheme, because really the only thing that's changed is not our understanding of the bacteria, but how we've named them. So we will cross that as we get to it, but just be aware, in this section, we're going through the new namings. So to start with, we're going to go through the diversity of microbes that are associated with the human body. We're going to leave out many of the environmental microbes, although I will give you one group that is a little bit of both. The other thing we're going to do is we're going to talk about a lot about big groups of gram positives, big groups of gram negatives, also some groups that don't fall into those broad categories there. And then we'll talk briefly, just lightly, about pathogen evolution and where we go look for microbes and why that is important. So let's start with our case study here. So we have Cody. Cody is 32 years old. He's from Fort Worth, Texas. And he is going out to the pub to shoot some pool. And as one does while shooting pool, you are required to have several beers. And then you are also required to get in a barroom brawl with his buddy Ty, who's also drunk, right? So these are all prerequisites of going to the pub and shooting pool. Obviously not. Don't get in barroom fights. But he goes for that little sock to the mouth there. And that makes short work of Ty. But what happens? Well, when you punch people, sometimes it hurts you. So again, we shouldn't be punching people, but also it can hurt you when you do it. So don't do it. More reasons. And he got a bite wound. And now this isn't like a chomp chomp bite wound. This is like a I hit you in the teeth and they broke my skin kind of wound, which happens. So again, don't get in fights. The thing about this is you probably don't notice, right? Your hand hurts. You got all kinds of other issues. You're drunk. You just had a fight with your buddy. You might not notice, but several days later, the wound starts to swell up and it gets red and it's warm to the touch, radiating heat. And it's really, really painful. So painful that Cody couldn't move his fingers. So here's a picture of this. We have swelling here. You can see gas forming under the tissue, these large necrotic areas of tissue, and you can see the redness here. And he has a fever overall of 37.9 degrees C. So 37 is our baseline, so we're elevated there. And his lymph nodes are not swollen here in this one. So what do we do? Well, first, we take an x-ray. We want to know, are there any broken bones in there? Maybe that's why he can't move his fingers. X-ray rules out broken bones. But we want to know what bacteria are in there, because obviously there's some sort of microbial infection going on there. And so we're going to take swabs of that wound and send that to the lab for culture to identify them. All right, so I want to talk here about a really important concept. This is the location of microbes and the body. So we have lots of microbes that are all over our body, and many of them, as we've said, are good parts of our microbiome. We often see problems, though, when microbes get out of place in the body. So in this case, we have results coming back, and we see what we call a polymicrobial infection. This polymicrobial means many different microbes, so there's a lot of different microbes in there. This includes mouth bacteria. So he did punch someone in the mouth, and some of the mouth bacteria got deposited in his wound, and skin bacteria. So the bacteria that were on his hand got shoved under the skin. And so we have like Eichinella rodens, Streptococcus pyogenes, Fusobacterium nucleatum, and Porphyromonas gingivalis there, a mouth bacteria. So we're seeing these normal bacteria that can cause some issues, minor issues usually, moving out of place. Once they get under the skin, they can cause large problems. This is a huge theme in microbiology. When things get out of place, bad stuff happens usually. So we diagnose him with what we call cellulitis, an infection of that soft tissue. It excludes muscle. That's the definition of cellulitis. And here, the mouth bacteria are really causing a serious infection. A physician would debride the wound, which means literally scraping off the dead tissue that's in there, cleaning it by irrigating with dilute hydrogen peroxide, and then we prescribe antibiotics, in this case amoxicillin, and maybe a tetanus shot as well. Tetanus vaccine works after infection, whereas most vaccines work pre-infection only. And we'll talk about why that is in a later chapter, in chapter 13. Okay, so we have a polymicrobial infection there. We need a broad spectrum antibiotic there to deal with that. All right, so let's dive into the naming here. We're going to do a quick overview of all the groups. Again, like I said, I'm going to use the new names now, but I'm going to use the older names in subsequent videos. Just because the material other than that hasn't changed. I will test you using the old names in the homework. If you see the new names, you're often also going to see the old names there. So you shouldn't have any problems. You can always refer back to this. And luckily, we didn't change the names too much there. So we're going to talk about 8 major groups of bacteria here. And we'll talk about a little bit about each, and then in subsequent sections of this chapter, we'll go into detail into each of those. Now, I want you to know I'm going to use some specific examples here, but in like testing and things like that, I will not pull out random examples that we didn't cover in great detail, or I've not made abundantly clear that they would be tested on. So you don't have to memorize every name of every species in there, but you might want to think about some of the big groups here. Make sure you know the 8 big groups that are in here. That is a definite must, and a little bit about them. All right, so we are going to talk quite a bit about pathogens, which are microbes that cause disease or can cause disease. We'll even talk about how new pathogens might emerge, but we'll also briefly talk about some positive roles of bacteria. I know this is a human medicine focused thing, so there are a few like microbiome examples and things like that, but I'm gonna stray outside and talk a little bit about environmental and ecosystem microbiology here. So here's our big phylogenetic tree of bacteria. And eukaryotes and archaea are off on their own over there. You'll see there are groups that we're not going to talk about. These are interesting microbes like aquifix. You can find that in Yellowstone. Not relevant to us. These often get called the deep branching thermophiles or things like that. What we're going to focus on mainly is in here. But I want to point out that since I taught this last, there is a new kind of group that's becoming recognized, and that is the candidate phyla radiation. So basically what this means is, hey, we're proposing that we think there's a new group over here, and there's a bunch of diversity in there. Most of these organisms weren't even discovered until starting in about 2007, and we've continued to gather evidence to support them. Now, the problem with this group is we don't know how to grow it. We think it could represent something like 15% of all bacteria species, but we can't grow them. Our evidence for them comes from DNA sequencing. We go out and we sample water and stuff like that, and the ocean or soil and stuff like that, and we see DNA from bacteria there, but we don't see the bacteria themselves. Many of these are super, super small, really, really small bacteria, and they have weird metabolism, things like that. So this is a new group. We don't know if there are any pathogens in here. That's why this is really interesting, right? Could there be new evolving pathogens in this group that we don't even know about? Could that explain diseases that we don't understand? We're still learning about that. So check back in, 10, 15 years, and I'll have a better answer for you there. The other groups here, we have a bit better handle on many of them with good examples in there. So we're going to focus mainly in here. So we have things like the cyanobacteriota, formerly the cyanobacteria. Those are an environmental group. We'll talk about them. Then we have this big group, the gram-positives. Now there are technically kind of three phyla in here. We have the Mycoplasmatota, which used to be within this group Firmicutes. We've kind of separated it out now. The Bacilliota, which used to be called the Firmicutes, the Actinomycetota, which used to be called Actinobacteria, and these are all gram-positives. Now, the gram stain is not 100% in here because there has been organisms that lose their cell wall. So like these Mycoplasmatota, they don't stain quite the same because they've actually lost their cell wall in here. So while they are related to one another, they don't always quite fit the gram stain perfectly, but on the whole, these are gram positives. If we look under this line that I've added here, we see our other groups. And in here, we have the gram negatives, which make up a large group here. We have the Pseudomonadota, which used to be called the Proteobacteria. Very important group in here. We got our friend E. coli in there. There's some subdivisions in there that we're not going to worry too much about. These blue ones we're not going to focus on. We will talk about the Bacteroidota, which used to be called Bacteroidetes. And then, outside of this, we have some organisms that don't really fit the gram-positive, gram-negative. They have different cell walls, different evolutionary history. So we'll talk about the Spirochetota, which are the spirochetes. And we'll talk about the Chlamydiota, which is formerly called the Chlamydiae. Very interesting group in here. So mainly we're looking at gram-positive, gram-negative, but there are some what we call gram-variable, or they don't really fit the pattern there. So let's look at the overall diversity here. Bacterial life is incredibly diverse. We have at least 30 different phyla. So I showed you 8 there. We have 30 different groups that are species that we can culture in the lab. Now, that might sound like a lot of stuff, but when we look at estimates of what's out there, we think there could be at least 1,000 or more phyla that we cannot grow. We have indications that they might be there, but we don't have any way of growing them, so we can't see them directly. Members of almost all of these 30 phyla are represented in the human microbiome. And we want to know more about microbial diversity in order to know more about what might cause disease. So let's take, for example, our E. coli, right? We said E. coli are a major player in the human microbiome. They help us digest our food. They're part of our gut. Well, how are they different from the toxic E. coli, the Shiga toxin producing E. coli? Another example, soil bacteria Bacillus anthracis, which causes anthrax disease, is toxic to humans. But the related bacteria, Bacillus thuringiensis, or BT, can be used as an insecticide. It will kill insects, but not humans. Why? We want to know about that, right? So the key is we got to group these things together so that we can then say, well, these two things are related. What makes them different, right? If we just go, okay, let's look at two vastly different things, we're going to have a harder time nailing down what causes the differences there. So in this section, we'll talk about 8 major superphyla groups that are known to impact human health and the ecosystem. So this is really focusing down on these. Again, in this section, I'm going to use the new nomenclature, the new naming. In subsequent sections, I will use the old naming. All right, so let's run through them here. In the gram-positives, we have 2 groups here. We have the Bacteriota. These used to be called firmicutes. We'll see in the later section why they have that name. And we have the Actinomycetota here, which used to be called the Actinobacteria. Really important, the Bacteriota, they often include organisms that can form endospores. Endospores are highly resistant structures that can resist chemical cleanings, heat, things like that. So this drying out, this makes them very difficult as pathogens to deal with. The Actinomycetota, on the other hand, are a really interesting group because they're where many of our antibiotics come from. Now that might seem strange, right? Antibiotics kill bacteria. Why are bacteria making antibiotics? Well, to compete with their neighbors, right? We got to remember that ecology still happens at the microbial level. So these bacteria have started making toxic chemicals that will kill their neighbors, basically, to compete for resources. Now, technically kind of in the group gram-positives, although they don't actually have a cell wall anymore, they've lost it, are the Mycoplasmatota here, which used to be contained within the firmicutes. We've broken them out into their own group here to add to that. So these bacteria, they're really, really small, simple self-replicating bacteria. They're some of the simplest out there. And like I said, they evolved from the Bacteriota, but they've subsequently lost their cell wall. So that happens in evolution. Sometimes we lose things. All right, so there's 3 groups. Okay, let's look at some more. Gram-negatives. We got 2 here. First off, we have the Pseudomonadota here, which used to be called Proteobacteria. Very important group. And then we have the Bacteroidota here, which used to be called Bacteroidetes. Pseudomonadota, very diverse in there. Basically, you have huge differences in how they do metabolism, create energy, things like this. But also in there, some very important organisms, particularly E. coli, which is very important in scientific research. We know so much about not just bacteria, but ourselves from studying E. coli. There's a famous quote by a microbiologist named Monod. He said, what's true for E. coli is true for the elephant. And he's speaking molecularly, right? Like they do glycolysis, the elephant does glycolysis. And by extension, we do glycolysis. So we've used them as a model system. And they're very important in biotechnology. The Bacteroidota, on the other hand, they're all gram-negative rods. Most of them are obligate anaerobes. That leads to some interesting things about where they live in us. They'll live in our gut tract, and many of them help digest our food. Okay, so we're up to 5 total here. We got 3 more. Okay, the Spirochetota here, used to be called the spirochetes. They have a very distinctive form to them. They have this tightly coiled cell in a kind of a corkscrew shape. It's enclosed by this sheath structure with a flagella that kind of runs along the inside of that, and they move in a corkscrew manner, do some interesting things, interesting diseases in there. Cyanobacteriota. Now this one, the cyanobacteria, this one's an interesting group because we talk about them. There are a few that are involved in disease processes, but most of them are out there doing photosynthesis. So they're the only bacteria that we know of that produce oxygen through photosynthesis. So they're really, really important for us because they're where most of our oxygen actually comes from. We like to think it's trees and go hug a tree, right? No, hug a cyanobacteria because that's where most of the oxygen's actually coming from out there. And then finally, we'll talk about the chlamydiota. The chlamydiae, formerly, these are obligate intracellular pathogens. That means they only grow inside of other cells. And we will see that that can be problematic for us because the sexually transmitted infection, chlamydia, is caused by these organisms here. So if we orient ourselves on the tree here, right, we have the Mycoplasmatota, which used to be in the firmicutes, but here we have the Bacilliota, which used to be the firmicutes. So we've broken these two groups up. They're in the gram-positives, and then an Actomycetota there, another gram-positive. Pseudomonadota here, formerly the Proteobacteria in the gram-negative, and the Bacteroidota here, which used to be called Bacteroidetes. Outside of that group, the Spirochetes and the Chlamydiota here are our groups. All right, to finish up here in this section, let's talk a little bit about microbial evolution and how potentially pathogens evolve. So I want you to remember back to last chapter, in chapter 9. We talked about mechanisms of bacteria sharing genetic material, right? So transformation and transduction and things like this. Well, new pathogens, things that can be disease-causing to us, are going to come from basically three places. They could be microbes that are out there in the environment, and they gain genes from other microbes that allow them to become pathogenic. In other cases, they are found in animals that we interact with, and we have an event called a zoonosis, where animal diseases can then infect humans. That's quite common. We have cowpox, which is a smallpox-related thing that infects cows, but can also infect people as they gain new genes, or we come into contact with them. These microbes can move to us, which is scary. Then we have this idea of shelter species. These are organisms that we don't necessarily directly interact with, but they give bacteria a place to live and interact and evolve together. And so, again, I want to reiterate this horizontal gene transfer. We have many microbes sharing bits of DNA, and that can lead to new pathogens evolving there. So this is always happening. Whenever microbial species interact, that can lead to new evolution occurring. Now, it seems all doom and gloom, and I don't want to end this chapter on doom and gloom, because humans can also impact this evolution and diversity, and sometimes we do that in interesting ways. For example, let's take bioremediation. Bioremediation is the idea that we could use living things to basically clean up toxic spills or things like that. So could we engineer microbes or help their evolution so that they can better break down like oil or something like that? We have an oil spill. There are microbes out there that can digest petroleum. They use it as food. What if we could help them along and direct their evolution to make them more efficient at doing that? We could clean up toxic spills or things like that. The idea of phage therapy, using viruses that kill bacteria to kill bacterial infections instead of antibiotics. This is an interesting topic. I'll talk briefly about it in the virus chapter. I think that's chapter 12. Then we have synthetic organisms. Scientists have actually made new DNA nucleotides not seen in nature before. Could we engineer completely new organisms with completely new DNA? Very interesting prospect. The idea probably would be to use those as microbial factories, right, to make various compounds and things like that and help prevent them from escaping because they have nucleotides that you can't find anywhere else. We have to give them to them. And then there's the specter of bioweapons, right? Really, what we need is to keep these from being made and or to learn how to defend against them. What makes microbes deadly and how do we prevent them from being deadly, or how do we counteract that is really the big question. So these are some positive aspects of microbial evolution, right? We have things like bioremediation, phage therapy, very interesting uses of the technology. All right, in the next sections, we will get into the individual groups.